On February 17, 1977, the submersible Alvin descended to the Galapagos Rift, expecting a cold, lifeless plain of basalt. Instead, its crew found dense colonies of giant tube worms, clams, and mussels thriving around warm, mineral-rich water seeping from the seafloor. Let the weight of the day drift away and let your body settle into the dark. We are going somewhere. There is time. The lights can stay dim. That single dive shattered the long-held belief that all life on Earth depends on sunlight. It revealed an entire kingdom of organisms fueled by the planet's inner heat, a discovery that rewrote biology. Today, these vents still exhale their black and white plumes in the deep, their chimneys glowing faintly, their strange inhabitants climbing blindly toward the warmth. For centuries, the deep ocean was imagined as a vast and silent void, a realm of perpetual darkness and crushing pressure. Scientists believed that beyond the sunlit surface waters, the seafloor was an unchanging, frigid desert almost entirely devoid of life. This view was not born of mere speculation, but was grounded in the limited observations of early naturalists, who saw no possibility for a complex ecosystem to exist without sunlight. The most influential voice behind this idea was the British naturalist Edward Forbes, who in the 1840s formulated what became known as the azoic theory. While dredging in the Aegean Sea, Forbes observed that the number of marine creatures declined steadily with depth, and he extrapolated this pattern to conclude that life would cease entirely below roughly 300 fathoms or 550 meters. His authoritative pronouncement cast the deep seafloor as a lifeless, primeval wasteland, a belief that took firm root in the scientific community. The landmark HMS Challenger expedition of the 1870s, which circumnavigated the globe and sounded the deepest trenches, delivered a profound challenge to the azoic theory. When its dredges returned from the abyss teeming with strange, fragile organisms, it proved beyond doubt that life could endure at depths exceeding 5,000 meters. Yet the sheer scarcity of these finds, spread across millions of square kilometers of ocean, failed to entirely overturn the notion of the deep as a biological desert. The discovery of life was acknowledged, but the general assumption of a profoundly impoverished world persisted well into the 20th century. By the mid-20th century, oceanographers had pieced together a picture of the abyssal plains as the most stable and monotonous environment on Earth. They expected to find a flat, featureless landscape of soft, brown sediment, accumulating at a rate of just a few millimeters every 1,000 years. The inhabitants of this realm were anticipated to be a sparse scattering of small, slow-moving deposit feeders, such as sea cucumbers and brittle stars, eking out an existence on a thin rain of organic detritus. The fundamental reason for this bleak assessment was the absolute absence of sunlight, which banished all photosynthetic life and left the deep sea in a state of perpetual cold, typically hovering just above freezing. Without plants or algae to form the base of a food web, complex life was thought to be impossible. The only energy source was marine snow, the gentle descent of waste and decay from the sunlit surface, and this meager supply was believed to be far too diffuse to support anything but the most frugal and isolated organisms. The immense pressure, which increases by 1 atmosphere for every 10 meters of depth, and the near-freezing temperatures were seen as additional barriers that would slow metabolism to a crawl. It was widely accepted that the deep ocean was a place of biological austerity, where life's processes were attenuated to the very edge of sustainability. The expectation, therefore, was not of vibrant communities, but of a delicate, almost invisible film of life, forever on the brink of starvation in a world of absolute darkness. In the early 1970s, the French-American mid-ocean undersea study, known as Project Famous, had proven that scientists could explore the deep ocean floor with remarkable precision. Using the French submersible Archimede and the American Alvin, the team successfully mapped and sampled a section of the mid-Atlantic ridge, confirming the processes of seafloor spreading and volcanic activity at the heart of a tectonic boundary. The mission demonstrated that direct human observation was not only possible but essential for understanding the raw geology of the rift. It was a triumph of collaboration that set the stage for a much more ambitious venture. When a new expedition was assembled to explore the Galapagos Rift in 1977, the primary objective was purely geological. The site, located where the Nazca and Cocos plates are pulling apart, promised a pristine laboratory for studying the mechanics of seafloor spreading and the chemistry of newly formed oceanic crust. Scientists on board the research vessel Cunora planned to tow deep-sea cameras, dredge rock samples, and deploy the submersible Alvin to examine fresh lava flows. The search for life was not part of the mission plan. The abyssal plain was considered a cold, dark desert, where only a few scattered organisms survived on a slow rain of organic debris from above. The vessel chosen for the deepest dives was Alvin, a white-hulled submersible operated by the Woods Hole Oceanographic Institution. It carried a crew of 3, typically a pilot and 2 scientific observers, inside a titanium pressure sphere barely 2 meters in diameter. The craft was rated to descend to a maximum depth of 4,500 meters, which put the entire Galapagos Rift within its reach. Its external manipulator arms, still cameras, and a basket for collecting samples made it a versatile platform, but the descent itself was a slow, cold, and silent journey through total darkness. On February 17, 1977, geologist Jack Corliss and geochemist John Edmund squeezed into the sphere alongside Alvin pilot Jack Donnelly for dive number 713. As the submersible sank through the water column, the deepening blue outside the viewports faded to an absolute black, and the temperature inside the sphere dropped to a damp chill. The descent took more than an hour and a half, during which the crew could do little more than conserve battery power and wait for the sonar to signal the approaching seafloor. Their target was a narrow fissure at a depth of roughly 2,500 meters, a place where they expected to find a monotonous terrain of pillow basalt. When the submersible's lights finally swept across the bottom, the first thing the observers noticed was a faint shimmering in the water, a distortion like heat rising from a summer road. This was the visual signature of hydrothermal fluids, buoyant and warm, mixing with the near-freezing seawater. Corliss and Edmund, trained to see rock, realized they were looking at a plume of hot, mineral-laden discharge venting from the seafloor. The temperature gauge on Alvin spiked as the craft drifted into the edge of the plume, confirming that they had stumbled upon something entirely unexpected and far from the cold, inert landscape they had anticipated. As the lights adjusted to the haze, the shimmer resolved into a vast and improbable congregation of living creatures. Clusters of giant white tubes, later identified as Riftia pachyptala, stood upright like a thicket of alien stalks, their crimson plumes retracting slowly at the disturbance. Dense accumulations of large clams and yellow mussels covered the basalt near the vent openings, and white crabs picked their way among the mats. Every single organism visible through the porthole was thriving in the complete absence of sunlight, sustained by a new kind of ecosystem built on the chemical energy seeping from the planet's interior. The submersible Alvin descended through the permanent darkness of the Galapagos Rift, its floodlights cutting a narrow cone through the near-freezing abyssal water. On February 17, 1977, the crew was surveying a region of volcanic seafloor spreading when they came upon a landscape that defied all biological understanding. Clustered around a crevice in the basalt were dense thickets of ghostly white tubes, some standing taller than a person. These were the giant tube worms, later named Riftia pachyptala, their vivid red plumes retracting slowly as the submarine approached. They had no mouth, no gut, and no anus, yet they thrived in colonies so dense they obscured the rock entirely. The largest specimens reached nearly 2 meters in length, their chitinous tubes swaying gently in the current of warm water rising from the vent. Scattered among the worms were enormous white clams, Calyptogena magnifica, some measuring 30 centimeters across. Their shells gaped open, revealing fleshy mantles that gleamed under the lights. Crawling over the clumps of mussels and across the sulfide crusts were blind white crabs, Bythogrea thermodron, their pale carapaces lending them an almost spectral appearance as they picked their way through the oasis. The external temperature probe registered a startling anomaly, water shimmering at 17 degrees Celsius, a profound contrast to the 2 degrees Celsius ambient temperature of the deep sea. The scientists inside Alvin stared through the viewport in total shock, their instruments and training having given them no framework for such a profusion of life in the absolute absence of sunlight. This was a community that could not exist according to every known rule of ecology. Geologist Jack Corliss, the dive's chief scientist, reached for the acoustic telephone and radioed the surface support vessel. He requested that a biologist be brought down immediately because what they were witnessing was not a geological phenomenon, but a biological revelation. The crackling voice that answered from the research ship Lulu could not conceal the bewilderment of the crew topside. In later recollections, the men inside that titanium sphere described the scene as nothing less than the discovery of the Garden of Eden. The comparison was not made lightly, for here, at the bottom of the ocean, was a self-contained world of abundance, powered not by the sun but by the Earth's own internal chemistry. It was a moment that rewrote the textbooks overnight, revealing that life could flourish in places once thought utterly barren. Hidden beneath the world's oceans, winding across every major basin, lies the longest mountain chain on Earth. The mid-ocean ridge system stretches for roughly 65,000 kilometers, a continuous volcanic seam where the planet's tectonic plates are slowly pulled apart. In the permanent darkness of the deep sea, this immense geological feature remains largely invisible from the surface, yet it is the very engine of ocean floor renewal. At these spreading centers, tectonic plates move away from each other at rates that can be as slow as the growth of a human fingernail, or as fast as several centimeters per year. As the plates separate, the rigid lithosphere thins and fractures, creating a network of deep fissures and faults. Molten rock from the mantle rises to fill the widening gap, solidifying into new oceanic crust and perpetuating the ridge's quiet, relentless construction. Seawater, cold and heavy, percolates downward through the shattered crust along thousands of tiny cracks. This slow descent can carry the water several kilometers into the ocean floor, far beyond the reach of sunlight. The downward journey is driven by the immense pressure of the overlying water column and the porous, brittle nature of the freshly formed rock. At depth, the descending seawater encounters magma chambers and newly solidified but still scorching hot rock. Temperatures can soar beyond 400 degrees Celsius, yet the immense pressure prevents the water from boiling. In this superheated state, the water becomes chemically aggressive, leaching metals and other compounds from the surrounding basalt, transforming into a fluid rich in dissolved minerals and hydrogen sulfide. The heated fluid, now less dense than the surrounding cold seawater, begins a rapid ascent back toward the seafloor. It finds focused pathways along larger fractures, eventually erupting from chimney-like structures in shimmering plumes. When this superheated, mineral-laden fluid hits the near-freezing abyssal water, the dissolved metals instantly precipitate, building the iconic dark spires and billowing clouds of black smoke that mark a hydrothermal vent. These vents are not scattered randomly across the ocean floor. They exist only where the geological conditions align, primarily along the actively spreading sections of mid-ocean ridges, as well as behind volcanic arcs and near mantle hotspots. The essential combination is a shallow heat source, such as a magma chamber, and a permeable crust that allows a full circulation loop of seawater to descend, be heated, and rise again. Without this precise tectonic setting, the deep sea remains a cold, quiet desert. Deep beneath the ocean surface, where tectonic plates pull apart, seawater seeps into the fractured crust and is superheated by the magma below. This superheated water, laden with dissolved metals and sulfide minerals, rises rapidly back toward the seafloor. When it erupts into the near-freezing ocean, the dissolved minerals instantly precipitate into fine dark particles, forming a rising plume that looks like thick black smoke. These particles settle around the vent opening, gradually building the hollow chimney structures we call black smokers. The fluids gushing from a black smoker can reach staggering temperatures of 400 degrees Celsius, hot enough to soften lead. At the seafloor, some 2 to 3 kilometers down, the immense weight of the water column creates pressures over 200 times that at the surface. Under such pressure, water does not boil even at 400 degrees, remaining in a liquid state that can carry an extraordinary load of dissolved minerals. It is this superheated, mineral-rich liquid meeting the frigid deep ocean that generates the characteristic dark plume. One of the most studied sites on the planet is the TAG hydrothermal field, located on the mid-Atlantic ridge at a depth of over 3,600 meters. Discovered in 1985, TAG stands for Transatlantic Geotraverse, and it hosts a large active mound made of massive sulfide deposits, with clusters of chimneys venting fluids at varying temperatures. The area has become a natural laboratory for understanding how these extreme environments sustain life and shape the ocean floor over millennia. The tallest known chimney ever documented was the Godzilla vent on the Juan de Fuca Ridge in the Northeast Pacific. This extraordinary structure rose 45 meters from the seabed, comparable to a 15-story building, before it was observed to have collapsed in 1995. Its towering, multi-spired form was a dramatic reminder that these edifices are built in a constant contest between rapid mineral growth and the forces of gravity. A chimney grows as mineral particles, primarily iron and copper sulfides, accumulate around the rim of a vent orifice. The fast-flowing fluid deposits layers of anhydrite and metal sulfides on the inner walls, insulating the conduit and allowing the structure to rise, often sprouting delicate branches and flanges. Growth can be surprisingly swift, with some chimneys adding several centimeters to their height in a single day. Yet these formations are inherently fragile. Veins of less stable minerals can dissolve, and blockages within the narrow conduit may divert the flow until pressure builds and the chimney fractures or implodes. Eventually, the sheer weight of a tall spire, combined with seismic tremors or the weakening of its base, leads to a dramatic collapse. The fallen fragments then become the foundation for a new cycle of growth, as mineral-rich fluids immediately begin to build again from the rubble. At the TAG hydrothermal field on the mid-Atlantic ridge, black smoker vent fluids reach 364 degrees Celsius. Deep beneath the Atlantic, far from the volcanic fury of the mid-ocean ridge, there exists a realm of gentler hydrothermal activity. These are the white smokers and diffuse flow vents, the quieter expressions of the planet's inner heat. Unlike their more dramatic black smoker cousins, they do not roar superheated fluids into the freezing darkness. Instead, they whisper, their warm fluids seeping slowly into the ocean, building landscapes of pale carbonate rather than dark metal sulfides. The most extraordinary of these hidden worlds is the Lost City hydrothermal field, discovered in the year 2000 by oceanographer Deborah Kelly during a deep-sea survey. It rests on the Atlantis Massif, a mountain of rock from the Earth's mantle thrust up to the seafloor. The moment her cameras glimpsed the ghostly white pinnacles, some towering over 60 meters, it was clear this was something entirely new. These were not the volcanic chimneys born of magma, but the spires of a city built by a different, ancient alchemy. The foundation of Lost City is a process called serpentinization, where seawater percolates deep into the exposed mantle rocks and reacts with the mineral olivine. This reaction, which generates heat without any magma at all, transforms the rock into slippery green serpentinite and releases hydrogen and methane into the fluid. When the warm, alkaline water rises and meets the cold ocean, dissolved calcium precipitates out, constructing the field's ethereal white chimneys and flanges from calcium carbonate, the same substance as limestone. The emerging fluids are a far cry from the acidic, metal-rich plumes of black smokers flowing out at a balmy 40 to 90 degrees Celsius with a pH as high as household ammonia. Moving away from these towering monoliths, diffuse flow vents paint an even more delicate picture. These vents do not erupt from a single orifice but seep from countless tiny cracks and fissures across the seafloor, like a warm breath exhaled through the skin of the planet. The flow is so slow and mixed with ambient seawater that it is often invisible to the naked eye, registering only as a subtle shimmer or a thermal gradient on a sensor. This gentle, pervasive emission creates a vast, low-energy habitat, where the boundary between the vent fluid and the ocean is a wide, soft gradient rather than a sharp, scalding divide. Chemistry and temperature define the boundaries of life here as strictly as any fence. The caustic, metal-saturated fluids of a black smoker demand specialist organisms like the giant tube worm, which relies on symbiotic bacteria that process hydrogen sulfide. The Lost City's alkaline, methane-rich waters, however, foster a different kind of microbial metropolis, dominated by archaea that thrive on the simple molecules of hydrogen and methane. You will not find dense thickets of tube worms on these carbonate spires. Instead, the chimneys are encrusted with thick, snotty biofilms, and the only visible animals are small, translucent snails and amphipods grazing upon this microscopic feast. These quieter vents offer a window into the deep, slow rhythms of the Earth, and perhaps the origins of life itself. The gentle, sustained reaction at Lost City has been active for at least 120,000 years, making it an oasis of stability in the abyss. Its cool, alkaline, and hydrogen-rich conditions bear a striking resemblance to the geochemical environments that many scientists believe may have cradled the first living cells. In the silent, patient seepage of these waters, the planet is not just venting heat but whispering a story of creation, one that continues to be written with every pulse of clear, warm fluid. Far below the reach of sunlight, across the abyssal plains and towering volcanic ridges of the deep ocean, Hydrothermal vents spew superheated water blackened with minerals. In this world of perpetual night, where pressures are immense and temperatures extreme, life does not merely endure. It thrives in astonishing abundance, powered not by the sun, but by a chemical alchemy born from the planet itself. The architects of this hidden abundance are chemosynthetic bacteria, single-celled organisms that harness energy from hydrogen sulfide gushing out of the Earth's crust. They draw in the sulfide, a compound toxic to most life, and combine it with oxygen dissolved in seawater. Through a carefully controlled oxidation reaction, the bacteria split the hydrogen sulfide molecules, releasing energy that they capture to convert carbon dioxide and water into simple sugars. The chemical equation is a fundamental act of creation, replacing a photon of light with a molecule of geological poison. In its function, this process is a perfect mirror of the photosynthesis that sustains the sunlit world above. Both pathways use an energy source to fix inorganic carbon into the organic building blocks of life. Yet the mechanism is entirely different, relying on a cascade of electron transfers that strip hydrogen from sulfide rather than splitting water molecules with light. It is a biochemical engine tuned to the geothermal heartbeat of the ocean floor, a revelation that the formula for life can be written in many energetic languages. These bacteria are the primary producers, the very base of the food chain around the vents. They form thick, filamentous mats that drape the rock in a living fleece, and they drift freely in the warm, mineral-rich water. Grazing animals such as limpets and amphipods scrape the mats directly, while filter feeders capture the drifting cells. Every creature in this ecosystem, from the smallest snail to the largest predator, depends on the continuous chemical harvest conducted by these microscopic alchemists. In a remarkable evolutionary twist, some vent animals have internalized this partnership entirely by housing the chemosynthetic bacteria within their own bodies. The giant tube worm, Riftia pachyptila, has no mouth, gut, or anus as an adult. Its bright red plume, feathery and rich with blood, absorbs oxygen and sulfide from the water, delivering both to a specialized internal organ called the trophosome. This organ is packed with billions of symbiont bacteria, a contained and protected farm that leaks sugars directly into the worm's bloodstream. This strategy of endosymbiosis is repeated across the vent landscape, with bathymodiolid mussels and vesicomyid clams harboring their own bacterial partners within the cells of their gills. The transparent shrimp of the mid-Atlantic ridge carry colonies of chemosynthetic bacteria on their mouthparts and scrape them for sustenance. Each partnership is a quiet testament to the intimate and enduring bargain struck between the animal and microbial worlds, a union that allows a sunless garden to flourish in the deep obsidian dark. In the abyssal darkness of the eastern Pacific, where volcanic heat escapes through cracks in the ocean floor, there exists a creature so improbable that its discovery rewrote our understanding of life itself. The giant tube worm, Riftia pachyptila, stands as the emblematic organism of hydrothermal vents, a white chitinous column topped with a plume of deep crimson that sways gently in the shimmering mineral-rich water. It can grow well over 2 meters tall, gathering in dense, silent congregations around the warm, sulfurous fluids that sustain an entire ecosystem without sunlight. What is immediately striking about Riftia is what it lacks. The adult worm possesses no mouth, no gut, and no anus. It is an animal that has completely abandoned the fundamental act of eating. Instead, deep within its body, a dark, spongy organ called the trophosome fills the majority of its trunk, packed so densely with bacteria that a single gram of the tissue can contain billions of symbionts. This organ is the worm's entire nutritional heart, a living factory where the bacteria convert vent chemicals into organic molecules that feed their host directly. The key to this internal alchemy lies in the worm's vivid red plume, which acts as a beautifully efficient exchange surface. The plume's color comes from an extraordinary set of hemoglobin molecules that circulate in the blood and also saturate the plume tissue itself. These hemoglobins have the unique ability to bind hydrogen sulfide absorbed from the vent fluid and oxygen drawn from the surrounding seawater, carrying both molecules simultaneously without letting them react prematurely. This carefully managed delivery system rushes the volatile cocktail down to the trophosome, where the bacteria safely oxidize the sulfide to fuel the synthesis of sugars and proteins. The partnership is so intimate that the worm is entirely dependent on its microbial tenants. The bacteria, chemolithoautotrophs, harness the energy released from the sulfide oxidation to fix inorganic carbon into the building blocks of life, much like a plant's photosynthesis, but powered by geology rather than a star. The worm provides the bacteria with a protected habitat and a steady supply of the raw materials they need, while the bacteria multiply and then are gradually digested by the worm's own cells. This complete internal cycling means Riftia lives without ever taking a single bite of food from the outside world. This way of life supports a rate of growth that is almost startling. Riftia pachyptila can lengthen its tube by up to 85 centimeters in a single year, making it the fastest growing marine invertebrate known to science. Newly formed vent fields, which can appear and vanish within decades, are rapidly colonized by these worms, their tubes racing upwards into the water column to claim the most favorable chemical gradients. It is a strategy of extreme speed in an environment where a thriving community can be extinguished overnight by a shift in the volcanic plumbing. Despite this fleeting boom and bust existence, the individual worm can endure for what seems an impossible span. By modeling the growth of their tubes over time, researchers have estimated that the largest Riftia individuals may live for over 250 years. This places them among the longest-lived of all invertebrates, silent sentinels that can persist through centuries of deep ocean quiet. Their existence is a paradox, a creature that grows with frantic haste yet measures its life in geological time, lifting its red plume into the darkness long after the vent that fed its youth has gone cold. In the permanent night of the deep ocean, hydrothermal vents roar with superheated water laden with minerals and chemical compounds. Here, life does not depend on the sun and photosynthesis, but on a different kind of primary production. Chemosynthetic bacteria, living freely in the water or within the tissues of animals, harness the energy locked in hydrogen sulfide and methane seeping from the seafloor. These microscopic organisms form the foundation of an entire food web, transforming the vent's toxic chemistry into living matter. Giant tube worms, with their vivid crimson plumes, rise in dense clusters from the basalt, lacking both mouth and gut. They are completely dependent on symbiotic bacteria that live deep inside a specialized organ called the trophosome. The worm's blood, rich in hemoglobin, transports hydrogen sulfide and oxygen to these internal bacteria, which in turn convert the chemicals into nourishment that sustains the host. Nearby, the vent mussel Bathymodiolus blankets the rocks, its gills packed with two types of symbiotic bacteria. Some oxidize sulfide, while others process methane, allowing the mussel to draw a double income from the vent's effluents. This dual symbiosis gives it remarkable resilience in the fluctuating chemical landscape. Scuttling among the mussel beds is the yeti crab, Kiwa hirsuta, a pale creature whose hairy arms appear surprisingly silken. Discovered in 2005, it cultivates dense gardens of filamentous bacteria on the long setae of its claws, rhythmically waving them through the oxygen-rich and sulfide-rich boundary layers to nurture its crop, which it then combs off and consumes. The darkness around the vents teems with millions of the shrimp Remicaris exoculata, swarming so thickly that they obscure the rock. These shrimp possess no ordinary eyes on stalks. Instead, they bear a pair of large reflective patches on their backs, unusual organs, sensitive to the faint geothermal glow radiating from the superheated fluids. This light is thought to guide them toward the vent source, while their specialized mouthparts scrape bacterial mats from the mineral surfaces. Gliding through the swarms with slow, deliberate movements, vent octopuses probe the crevices with their arms, seeking out shrimp and crabs. Eelpouts, slender fish with elongated bodies, drift in the cooler margins, moving in to snatch smaller crustaceans and worms. The predatory fish Thermarsis cerberus, a relative of the eelpout, hunts actively among the tube worm colonies, its jaws well suited to tearing at the soft bodies of invertebrates. Thus, the food web spirals outward from the vent's chemical bounty. The chemosynthetic bacteria feed the tube worms, the mussels, the shrimp, and the cultivated gardens of the yeti crab. These primary consumers in turn sustain the wandering octopuses, the vigilant eelpouts, and the hunting Thermarsis cerberus. In this oasis of the abyss, a complete ecosystem pulses with a slow, ancient rhythm, powered not by the sky, but by the planet's own internal fire. The iron-saturated plume billows like a curtain of rust underwater. A blind shrimp plucks bacteria from the cloud with the delicacy of a pianist's fingers. The 1977 expedition to the Galapagos Rift documented entire ecosystems thriving in permanent darkness, sustained not by sunlight, but by chemical energy seeping from Earth's interior. With that single observation, the long-standing assumption that all life depends on photosynthesis quietly fell away. It raised a quiet, persistent question. If life could flourish in such extreme isolation, where else might it be waiting, hidden in the deep ocean crust, beneath polar ice, or in the subsurface oceans of distant moons? Deep beneath the ocean surface, where tectonic plates slowly pull apart, towering mineral chimneys rise from the sea floor and release clouds of warm, mineral-rich water into the cold abyss. These are hydrothermal vents, and for decades they have been at the center of a profound scientific inquiry into how life on Earth began. Among the most compelling ideas is the alkaline hydrothermal vent theory, developed by geologist Michael Russell and biochemist Nick Lane. It proposes that life did not emerge in a sunlit tidal pool but in the dark, porous labyrinths of deep sea vents, where a natural chemical engine was already running. In the year 2000, the discovery of the Lost City hydrothermal field in the mid-Atlantic offered a glimpse of a world that may have been the cradle of biology. Unlike the black smokers that belch volcanic sulfides, Lost City's ghostly white carbonate spires form through a process called serpentinization, where seawater reacts with mantle rock to produce warm, alkaline fluids rich in hydrogen and methane. These towering structures, up to 60 meters tall, are riddled with microscopic pores, each a tiny mineral compartment that could have concentrated and protected the first organic molecules. The gentle, steady flux of chemistry here stands in stark contrast to the violent heat of volcanic vents, providing a far more hospitable nursery for the earliest steps toward life. At the heart of this theory lies a strikingly simple gradient. The alkaline fluid welling up through the vent chimneys has a pH of around 9 to 11, while the early Earth's carbon dioxide-rich oceans were mildly acidic, perhaps with a pH of 5 to 6. This natural disparity across the thin mineral walls of the vent pores created a proton gradient, a difference in the concentration of hydrogen ions, much like the voltage across a battery. Remarkably, this is the very same kind of electrochemical gradient that powers every living cell today, where mitochondria pump protons across their inner membranes to drive the synthesis of ATP, the universal energy currency of life. The vents, it seems, offered a natural prototype of the system that still fuels our own bodies. Inside those interconnected microchambers, the conditions were ripe for the emergence of complex chemistry. The mineral walls, composed of iron and nickel sulfides, can act as catalysts, encouraging simple molecules like carbon dioxide and hydrogen to react and form organic compounds. Within these labyrinths, the building blocks of RNA and the metabolic cycles that sustain life could have been born together, not in a sequential chain of improbable events, but in a continuous, self-reinforcing process. The pores served as a natural concentrating mechanism, trapping and preserving molecules long enough for them to interact, replicate, and eventually give rise to a primitive RNA world, all while the proton gradient provided a steady drizzle of energy. A growing number of scientists now regard the deep sea vent scenario as more plausible than the classic image of Darwin's warm little pond. A surface pond, however rich in organic soup, would have been exposed to intense ultraviolet radiation, evaporation, and the diluting effects of rain, making the sustained assembly of complex molecules difficult. In contrast, the vent environment offered a continuous supply of chemical energy, a protected three-dimensional compartment, and a thermodynamic gradient that could drive the formation of ever more complex structures. The pond relied on chance lightning strikes and drying cycles for concentration, while the vent provided a steady, reliable, and deeply resonant chemical pathway from geochemistry to biochemistry. The alkaline hydrothermal vent hypothesis thus paints a picture of life's origin as a gradual, almost inevitable transition from rock to cell. It connects the slow churning of the planet's interior to the delicate biochemistry that defines every living thing, suggesting that the first spark of life was not a sudden flash but a gentle, sustained current. Deep in the dark, silent chambers of a lost city-like vent, the foundations of our existence were quietly laid, molecule by molecule, in a microscopic garden of mineral towers. Deep beneath the ocean's surface, hydrothermal vent fields rise from the abyssal plain like submerged archipelagos. Each field is a cluster of mineral spires separated from the next by vast stretches of cold, barren seafloor. The distances between them, often hundreds of kilometers, make these habitats as isolated as volcanic islands on land. They are oases of chemical energy in a dark desert, and the creatures that live here are found nowhere else. The animals inhabiting a vent field are the descendants of larvae that once arrived on deep ocean currents. Because the currents are sluggish and larvae survive only for a limited time, successful dispersal to another vent is a rare and chancy event. Most larvae settle close to their birthplace, reinforcing the genetic separation of each population. Over generations, this isolation allows each group to evolve along its own independent trajectory. Within these isolated pockets, evolution proceeds at a remarkably accelerated pace. The extreme gradients of temperature and chemistry, along with the high metabolic demands of chemosynthetic symbiosis, exert intense selective pressure. Vent species, such as the deep sea snail Alviniconcha, accumulate genetic changes far faster than their relatives in shallow, sunlit waters. The result is a rapid proliferation of new species, each adapted to the peculiar conditions of its own vent field. When a fresh volcanic eruption creates a new vent system, the bare rock is first colonized by a bloom of free-living microbes. These bacteria and archaea coat the surfaces in thick, glistening mats, feeding directly on the dissolved minerals and gases. Their presence transforms the rock into a living substrate, paving the way for more complex organisms. Within a few years, the microbial mats are joined by the first wave of animals, typically tube worms, limpets, and amphipods. The tube worms, which cultivate symbiotic bacteria within their bodies, grow rapidly and form dense bushes that create a three-dimensional habitat. This structural complexity attracts mussels, crabs, and swarms of shrimp, and over time the community matures into a stable ecosystem. The succession from microbes to a full animal community mirrors the recovery of a forest after a fire, compressed into a handful of years. The existence of a hydrothermal vent is fleeting on a geological scale. When the subterranean heat source shifts or the plumbing is sealed by mineral deposits, the flow of superheated water ceases. The animals, utterly dependent on chemical energy, perish. Yet extinction is not necessarily the end. The dying vent releases a final pulse of larvae into the currents. These tiny voyagers drift through the abyss, and for the few that chance upon a new active vent, the cycle of isolation and evolution begins again. In the eternal darkness of the deep ocean, where sunlight never penetrates, lies a world that was entirely unknown to science until 1977. That year, researchers exploring the Galapagos Rift aboard the submersible Alvin discovered towering chimneys spewing superheated, mineral-laden water into the cold abyss. Around these hydrothermal vents, an astonishing ecosystem thrived in absolute darkness, powered not by photosynthesis but by chemosynthesis. The discovery fundamentally rewrote the rules of biology, proving that life could flourish on chemical energy alone, entirely independent of the sun. Quietly, it redrew the boundaries of the habitable zone, extending it far beyond the gentle warmth of a star's light. This revelation cast a distant moon of Jupiter in a new and tantalizing light. Europa, a world of cracked and icy plains, conceals a global ocean of liquid water beneath its frozen shell, kept warm by the relentless gravitational kneading of its parent planet. The ocean floor is not a sealed, dead tomb, but a rocky seafloor in direct contact with the water, creating the perfect conditions for water-rock interactions. The telltale signs of tidal flexing around Europa's surface suggest that magma may rise from the mantle, potentially fueling hydrothermal vents on the ocean floor. In the profound silence of that dark, salty sea, the same chemical duets of hydrogen and carbon dioxide that sustain life on Earth could be quietly playing out. 500 million kilometers further out, Saturn's small moon Enceladus has been caught in the act of revealing its own secrets. The Cassini spacecraft flew through enormous geysers of water vapor and ice erupting from the moon's south polar region and sampled the spray. Within those ethereal plumes, instruments detected tiny grains of silica nanoparticles of a specific size and purity that can only form in water that is superheated and moderately alkaline. The analysis pointed to active hydrothermal vents on Enceladus's seafloor, with temperatures exceeding 90 degrees Celsius. Here, on a tiny moon barely wider than the length of the United Kingdom, the ocean floor is actively cooking the deep water, generating the kind of energy gradient that life can tap into. The discovery of the Galapagos vents did more than just add a new habitat to Earth's inventory. It quietly dismantled the long-held assumption that a habitable zone was defined solely by a planet's distance from its star. Astrobiologists now understand that a world can be utterly frozen on the surface and yet cradle a liquid water ocean in its interior, heated by tidal friction or radioactive decay. The classic habitable zone, the Goldilocks region where water can exist as a liquid on the surface, is now complemented by a vast, subsurface habitable zone that extends to the outer reaches of any given solar system. A moon like Europa, orbiting far beyond the reach of the sun's gentle heat, suddenly became a prime candidate for life. This is why hydrothermal vent environments are now considered among the most promising targets in the search for extraterrestrial life. They offer a self-contained, stable habitat, shielded from the harsh radiation and vacuum of space by kilometers of ice and rock. The chemical energy that pours from these vents is not fleeting. It is a continuous, reliable power source that can sustain complex ecosystems for millions of years. An origin of life at a deep-sea vent, a scenario actively studied on Earth, would be naturally protected from the sterilizing ultraviolet light and cosmic impacts that plagued the early surface of any planet. In the quiet, dark nurseries of a seafloor, the first fragile steps of biochemistry could proceed undisturbed. The search now moves slowly, deliberately, towards these distant ocean worlds. Missions like Europa Clipper are designed not to land, but to orbit and perform detailed reconnaissance of the moon's ice shell and what lies beneath it, tasting the faint traces of ocean that might puff up through cracks. The evidence from Enceladus, a plume sample already in our hands, provides a chemical fingerprint of its ocean floor so compelling that missions to return to that tiny moon are being dreamt into existence. The deep, dark boundary where hot rock meets cold water is no longer a niche curiosity of Earth's ocean floor. It is a quiet cosmic engine, a place where the whispers of life might be stirring in the long, patient night of the outer solar system. Hydrothermal vents are scattered across every ocean basin, forming a hidden network of black smokers, white chimneys, and diffuse flow fields. On the mid-Atlantic ridge, the Rainbow and Lost City vents sit in the slow-spreading crust, while the East Pacific Rise hosts the fastest known vent fields, such as 9°50 north, where continuous eruptions reshape the seafloor. The Juan de Fuca Ridge off the coast of Oregon and Washington supports the Endeavour segment, a cluster of towering spires, and the Indian Ocean Ridge holds the Kyre and Edmund fields, whose fluids are enriched in metals and hydrogen. Even the Southern Ocean around Antarctica harbors vents on the East Scotia Ridge and the Bransfield Strait, where life endures in near-freezing darkness. Discovering a new vent field begins with detecting anomalies in the water column above the seafloor. When superheated fluid erupts from a vent, it mixes with cold seawater, creating a plume that is warmer, more acidic, and laden with dissolved metals and particles. Research vessels tow conductivity temperature depth sensors, or CTDs, through the water, looking for sharp spikes in temperature, turbidity, and chemical signatures such as methane and hydrogen sulfide. These plumes can rise hundreds of meters before dispersing, acting as a trail that reveals the chimney below. To pinpoint the exact location, scientists deploy autonomous underwater vehicles, or AUVs, that glide close to the bottom. These vehicles carry side-scan sonar and magnetometers, but they also map heat signatures using high-resolution temperature probes and chemical sensors. The AUVs fly in a lawnmower pattern, sniffing the water for the faintest thermal anomaly, and when they encounter a plume, they can follow it back to its source. The result is a precise map of the vent field, often revealing structures that are invisible to shipboard sonar. All confirmed vent fields are cataloged in the Interridge Global Database of Active Submarine Hydrothermal Vent Fields, which is maintained by an international community of scientists. As of the latest update, the database lists just over 700 known vent fields, each with a unique identifier, location, depth, and a description of its geology and biology. Yet this number represents only a fraction of the world's active vents, because the global ocean floor spans more than 360 million square kilometers, and most of it remains unexplored. Estimates suggest that thousands of vent fields still lie undiscovered, hidden in remote ridges, back arc basins, and poorly mapped seamounts. The slow, spreading ridges of the Atlantic and Indian Oceans are particularly undersampled, and the Arctic Ocean has only a handful of confirmed vents. Each new discovery expands our understanding of Earth's geothermal processes and the extreme ecosystems that thrive in them. The search for these hidden oases continues, driven by expeditions that combine satellite gravity data, global ridge mapping, and the persistent work of research vessels and AUVs. Every anomaly detected in the water column is a clue. Every heat signature a promise of a new vent field waiting to be seen. The deep ocean still holds most of its secrets, and the hydrothermal vents are among the most compelling. Far below the reach of sunlight, where the Earth's crust fractures and superheated water billows into the abyss, hydrothermal vents build towering mineral cathedrals. These black smokers and their diffuse flow counterparts slowly deposit polymetallic sulfide structures rich in copper, zinc, gold, and silver. The deposits, formed over thousands of years, have drawn the attention of a nascent deep-sea mining industry that sees them as a solution to the growing demand for metals used in electronics and green technologies. The International Seabed Authority, established under the United Nations Convention on the Law of the Sea, holds the mandate to regulate mineral-related activities in the international seabed area beyond national jurisdiction. It has issued exploration contracts for vast tracts of the ocean floor, including zones along the mid-ocean ridges where hydrothermal systems are concentrated. The authority is now in the process of developing a comprehensive exploitation code, a set of rules that would govern how commercial mining could proceed, balancing resource extraction with the protection of the marine environment. The ecological argument against mining these vent fields is rooted in the extraordinary life they support. Active vents host chemosynthetic communities that exist nowhere else, from dense colonies of giant tube worms and shimmering mussels to blind shrimp and extremophile microbes. The argument extends to so-called inactive vent fields, where the hot fluids have ceased to flow. Research suggests these sites are not barren graveyards, but rather serve as crucial stepping stones and refugia for deep-sea species, and their massive sulfide structures provide habitat for slow-growing corals, sponges, and other organisms that may take millennia to establish. A single mining operation, designed to cut and scoop the seafloor, would strip away the entire three-dimensional architecture of a hydrothermal edifice. It would destroy not merely an isolated cluster of animals, but an entire ecosystem that has assembled over centuries of vent community development. The slow pace of growth and the episodic nature of vent activity mean that recovery is measured in human lifetimes, if it occurs at all. The sediment plumes generated by the cutting machinery could smother filter-feeding communities far beyond the mine site, carrying the disturbance across deep ocean currents. These concerns have fueled a growing international discussion around a moratorium on deep-sea mining. Several nations, along with a coalition of indigenous groups, civil society organizations, and global financial institutions, have called for a precautionary pause until robust scientific evidence can demonstrate that exploitation will not cause irreversible harm. The negotiations at the International Seabed Authority have become a focal point for this debate, with some member states pushing for a swift adoption of regulations, while others insist that no mining should begin without a clearer understanding of the consequences. The scientific community has articulated its position with increasing clarity through peer-reviewed studies and consensus statements. Marine biologists and deep-sea ecologists warn that we are on the verge of potentially extinguishing unique life forms before they are even cataloged, losing ancient genetic libraries and biotechnological potential. The core of their message is one of profound, unanswered questions. We do not yet know enough about the connectivity, resilience, and baseline biodiversity of these vent systems to authorize their deliberate destruction, and the absence of knowledge should be reason enough to wait. In the absolute dark, where the weight of the ocean would crush us in an instant, life did not merely survive. It learned to drink the mineral breath of the earth itself, to build bodies from hydrogen sulfide and raw heat. The tube worms, the blind shrimp, the microbial mats that shimmer like forgotten cities deep in the stone all whisper the same secret. There is no place so hostile that a pulse of invention cannot find a foothold. You can feel it now, as a quiet certainty settling in your ribs. This is not a story about a distant seabed. It is the story of existence persisting where nothing should, a pattern woven into the fabric of every living thing. A seed can crack concrete. A single cell can hold the blueprint for a forest. And in the blackness beneath the crust, the same stubborn spark that lights the vents is the one that kept your own ancestors breathing through ice ages and long nights, a patience that predates light. Now, the image of those black smokers softens, their dark plumes rising like slow, temperatureless breaths in the deep. The water cools, and the dense, heavy silence wraps itself around the planet's hidden fires. Let your own breath find that rhythm, a gentle mineral exchange, a tide without a shore. The abyss is not empty, it is full, and it is dreaming. The deep ocean floor remains one of the least explored frontiers on Earth, and the hydrothermal vents that punctuate its vast plains are still full of unanswered questions. Every expedition to a new vent field reveals species unknown to science, yet the very act of discovery has historically been so slow that we have only glimpsed snapshots of these ecosystems. Now, the pace is accelerating dramatically, driven by a new generation of autonomous underwater vehicles, high-resolution seafloor mapping, and portable DNA sequencing that can be deployed at depth. These tools are allowing scientists to revisit the same sites with a frequency that was once unimaginable, opening a window onto the hidden rhythms of vent life. Despite this progress, remarkably few vent fields have been visited more than a handful of times over the decades. The harsh reality of deep-sea research, with its reliance on expensive ships and submersibles, means that most known sites were described during a single expedition and then left unmonitored for years. This snapshot approach leaves a fundamental gap in our understanding, because we cannot track how communities shift, age, or recover from disturbance without repeated observations. Time series data from a small number of well-studied sites, such as those on the Juan de Fuca Ridge, hint at complex cycles of growth and collapse. But for the vast majority of the planet's vent systems, the dimension of time remains entirely missing. One of the most persistent mysteries is how vent larvae, drifting as tiny specks in a lightless ocean, manage to find a new home on a chimney that may be tens of kilometers away. The larvae of tube worms, mussels, and shrimp must settle onto a suitable vent before their brief energy reserves run out, yet they are born into currents that should sweep them into oblivion. Scientists suspect they follow chemical gradients, perhaps sensing sulfide or methane, or even respond to the sound of venting fluids, but direct evidence of their navigational cues is still elusive. Understanding this larval odyssey is crucial because it underpins the resilience of these island-like ecosystems in the face of natural and human disturbance. Puzzling patterns are emerging from the genetic fingerprints of vent populations, adding to the intrigue. In some cases, communities on vents separated by thousands of kilometers of barren seafloor show a strange synchrony in their reproductive cycles or population booms, as if connected by an unseen force. This phenomenon, observed in tube worm settlements along the East Pacific Rise, defies simple models of larval transport, which would predict a much more haphazard and disconnected pattern. The synchrony hints at a subtle environmental trigger, perhaps linked to large-scale ocean currents or subtle shifts in vent chemistry that we are only beginning to perceive. When a volcanic eruption blankets a vent field in fresh lava, it extinguishes every living thing in an instant, creating a blank slate of sterile rock. The few sites where this has been filmed, such as the Nidogan vent field on the East Pacific Rise, show a remarkably rapid recolonization, with microbial mats appearing within days and a full animal community re-establishing in just a few years. Yet the precise sequence of who arrives first, how they alter the chemistry of the nascent vents, and what determines the final composition of the community remains a story told from only a handful of resets. Each eruption offers a rare natural experiment, but the window of opportunity to witness it is fleeting and logistically daunting. The deepest parts of the ocean, the Hadal trenches that plunge beyond 6,000 meters, hold a final, tantalizing secret. Geochemical calculations in sediment cores suggest that a different kind of venting, driven not by volcanism, but by the reaction of seawater with exposed mantle rock, should exist in these crushing depths. Such serpentinization-driven vents would be cooler, rich in hydrogen and methane, and might host life forms utterly distinct from the sulfide-reliant communities we know. As of now, no actively venting site has been visually confirmed in a Hadal trench, but the search is intensifying with submersibles now capable of reaching these extreme pressures, promising a discovery that could rewrite the rules of life on our planet. The view through Alvin's 3 small portholes shifts within minutes from the turquoise shimmer of the surface to a deep, inky blue. By 200 meters, the last traces of sunlight have dissolved and the world outside becomes a darkness so complete it feels almost solid. Inside the sphere, the only illumination comes from the soft glow of instrument panels and the occasional flash of bioluminescent creatures drifting past like green sparks in the void. As the submersible sinks further, the titanium hull creaks gently, a sound that veteran pilots recognize as the sphere adjusting to the immense pressure. At 2,500 meters, more than 250 kilograms of force press on every square centimeter of the exterior, and the internal temperature drops enough to make the 3 occupants reach for an extra layer. The silence at depth is a physical presence, broken only by the hum of the scrubber removing carbon dioxide from the air and the muted clicks of the acoustic telephone. The first glimpse of hydrothermal vent light is a moment most scientists struggle to put into words. It appears as a faint, ghostly shimmer on the seafloor, a place where the eye expects no light at all. This glow emanates from superheated, mineral-rich fluids meeting the near-freezing seawater, a phenomenon scientists attribute to thermal radiation and sometimes crystalloluminescence as minerals precipitate. One researcher described it as seeing the Earth breathe light. Even sealed inside the pressure sphere, the signature of the vents slowly intrudes. A sharp, unmistakable scent of hydrogen sulfide, reminiscent of rotten eggs, seeps through the air circulation system. This odor is the first sensory confirmation of the chemistry that sustains an entire ecosystem in the permanent dark, a process called chemosynthesis that relies on the very compounds that smell so pungent to human senses. The external temperature probes tell a story of eerie thermal gradients. In the fraction of a second between the probe's exit from the cold water and its entry into the shimmering plume, the reading can jump from 2 degrees Celsius to over 350. The submarine's frame registers this warmth as a subtle expansion, a tingle of data that translates on the pilot's readouts into a landscape of invisible heat bleeding from the ocean crust. Emerging into the vent field feels like arriving at a planetary boundary, a terrain that belongs to no known world. Carbonate spires rise from the basalt, their flanks encrusted with white tube worms and swaying blind shrimp. Every surface is a riot of life that has no reliance on the sun, and the realization settles in slowly that this is a place where the rules of the surface world simply cease to apply. Deep beneath the ocean surface, along the volcanic ridges of the seafloor, hydrothermal vents discharge fluids that are among the most chemically extreme environments on Earth. These fluids originate as seawater that percolates through fractured basalt, is heated by magma to temperatures exceeding 400 degrees Celsius, and undergoes a series of chemical reactions that strip it of oxygen and dissolve vast quantities of minerals. The result is a superheated, acidic brine with a pH that can drop to near 2, comparable to stomach acid or lemon juice, loaded with dissolved metals including manganese, iron, copper, zinc, and cobalt, as well as copious amounts of hydrogen sulfide. The acidity and metal content of vent fluids arise from the interaction of seawater with hot rock at depth. As the water is heated under immense pressure, sulfate in seawater is reduced to hydrogen sulfide, while the hot, oxygen-free fluid leaches metals from the surrounding basalt. The dissolved ions of iron, copper, zinc, manganese, and cobalt become concentrated in the fluid, which remains in a metastable state until it erupts back into the cold ocean. When the superheated vent fluid exits the seafloor, it meets near freezing seawater, typically around 2 degrees Celsius, just centimeters away from the vent orifice. The temperature gradient across this boundary is staggering, creating a thermocline so sharp that the 2 water masses barely mix. The fluid, though hot and buoyant, is immediately cooled, and its chemistry begins to shift dramatically. The rapid cooling and the sudden increase in pH cause the dissolved metals to precipitate out of solution in a precise cascade. Iron sulfide, or pyrite, forms first, followed by chalcopyrite rich in copper and iron, then sphalerite containing zinc, and later traces of cobalt and manganese-bearing minerals. These solid particles, dark and fine, give the black smoker its characteristic plume as they are swept upward. On the vent surface itself, the precipitation builds a towering chimney structure layer by layer. Anhydrite, a calcium sulfate mineral, precipitates first from the heated seawater surrounding the vent, forming a porous outer wall. Inside this shell, the metal sulfides accumulate, creating a dense, mineral-rich lining that can grow several meters tall over the course of a single year. The chemistry of vent fluids not only constructs these remarkable chimneys, but also sustains entire ecosystems of organisms that thrive on hydrogen sulfide and dissolved metals. The precise balance of temperature, pH, and mineral saturation at the vent margin dictates the structure of the chimney and the composition of the plume, making each black smoker a unique chemical laboratory in the deep ocean. In the profound darkness of the deep ocean, Hydrothermal vents are not silent monuments. They crackle with the sound of metal sulfides precipitating from superheated fluids, a faint percussive chorus as minerals flash into solid form upon contact with the icy seawater. Each tiny crystal formation adds a delicate snap to the vent's sonic signature. Beneath the seafloor, the planet's molten breath produces a low-frequency rumble that resonates through the vent structures. This infrasonic thrumming originates from magma chambers and the movement of fluids through subterranean conduits, a deep bass note that underpins the vent soundscape. It is a sound too low for human ears to perceive directly, but one that hydrophones translate into a steady, grounding pulse. Among the towering mineral chimneys, life adds its own voice. Swarms of vent shrimp, grazing on thick mats of chemosynthetic bacteria, produce a constant staccato of snaps. Their specialized claws collapse cavitation bubbles with astonishing speed, creating a crackling that rivals the sound of the mineral precipitation itself. This biological percussion enriches the acoustic texture, betraying the dense communities thriving in the toxic outflow. Hydrophones deployed on the seafloor have patiently recorded these vent soundscapes, revealing a sonic environment far richer than anyone imagined. The recordings capture the crackle of sulfide chimneys, the rumble of geothermal activity, and the ceaseless snapping of shrimp layered together into a unique acoustic fingerprint. These soundscapes allow scientists to monitor vent health and activity from a distance, without disturbing the fragile ecosystem. The same hydrophone networks that listen to vents also eavesdrop on the birth of new seafloor. Eruptions at mid-ocean ridges are often detected acoustically long before any visual confirmation is possible. The violent release of lava and superheated fluids generates a distinct seismic signature that travels through the water, alerting researchers to a seafloor eruption days or even weeks before a submersible can reach the site. This acoustic early warning system has transformed our understanding of deep-sea volcanism. The most far-traveling signatures of these submarine eruptions are known as T-waves or tertiary waves. They are acoustic energy that becomes trapped in the ocean's SOFAR channel, a natural sound conduit that can carry the signal across entire ocean basins. By analyzing the arrival times and frequencies of T-waves, scientists can pinpoint the location of an eruption and estimate its intensity, revealing a hidden map of volcanic activity along the globe's mid-ocean ridges. The ocean itself becomes a vast listening device, turning the deep sea's volcanic whisper into a planet-wide announcement. A black smoker vent field is born in a cataclysm of volcanism along the deep ocean ridge where superheated water, laden with dissolved metals and sulfide, erupts into the cold abyss. These chimneys can grow with astonishing speed, sometimes meters in a single day, yet their lifespans are surprisingly brief. Individual vents may remain active for a few decades to a few centuries before the plumbing is sealed by mineral deposition or the underlying heat source shifts. When the flow of hydrothermal fluid finally ceases, the collapse of the vent community is swift and absolute. The dense congregations of giant tube worms, blind shrimp, and bathymodiolid mussels, which depend entirely on chemical energy from the vent fluid, begin to perish within days to weeks. Within months, the habitat is stripped of its most iconic obligate species, their tissues consumed or decomposed, leaving only the mineral skeleton behind. The transition from a roaring black smoker to a quiet sulfide mound is a process of mineralogical and biological reclamation. The once hot acidic fluids no longer bathe the chimney walls, which begin to cool and weather in the ambient seawater. Over time, the fragile spires collapse under their own weight or through seismic tremors, forming a rubble-strewn field of polymetallic sulfides that slowly oxidizes to a rust-brown crust. Even in this apparent death, the inactive mound is far from barren. The porous rock continues to host a hidden microbial world, a cryptic biosphere that thrives on the residual chemical gradients. These communities are dominated by bacteria and archaea that can oxidize the iron and sulfur minerals within the dead chimney or consume the hydrogen and methane still diffusing faintly from the subsurface. Their metabolism is slow, their tempo measured in geological time, yet they sustain a simplified food web of protozoa and small invertebrates. To understand the cycle of a vent field, one must step back to the scale of ridge volcanism, which operates across tens of thousands to millions of years. The tectonic plates are in constant slow motion, and the magmatic heat engine beneath a vent field is a flickering migratory pulse. A single volcanic eruption can smother an existing vent community in lava, while simultaneously creating a new fissure system a few kilometers away, a fresh canvas for colonization. The entire vent field landscape is a mosaic of life stages, from newly born chimneys to ancient cold mounds. The old mounds, slowly dissolving and being buried by pelagic sediment, represent the final chapter in a vent's biography. They can persist for tens of thousands of years, becoming islands of hard substrate and microbial activity on the abyssal plain until the ridge's volcanic cycle may one day refresh them. A new intrusion of magma can reheat the buried plumbing, reactivating the flow and resurrecting a vent field at the same site, beginning the ephemeral, life-giving cycle once more. In the winter of 1985, a deep-sea expedition to the Transatlantic Geotraverse, or TAG, hydrothermal field on the mid-Atlantic ridge brought a most peculiar creature into the light of science. The remotely operated cameras emerged from the abyss, carrying images of dense, seething masses of shrimp, entirely new to human knowledge. This was Rimicaris exoculata, a species that would come to redefine our understanding of how life perceives its world in the permanent night of the deep ocean. The most striking oddity of Rimicaris exoculata lies in its sensory biology, a complete departure from the visual systems of its sunlit cousins. Its compound eyes, those multifaceted windows typical of crustaceans, are present in the larval stage but degenerate as the shrimp matures. In their place, a large fused dorsal organ develops like a brilliant white saddle across the back of the carapace. This is not a remnant but a highly derived photoreceptive organ, exquisitely tuned to the dimmest of light sources, a biological photometer staring up into the void. The source of that light is not the distant sun, but the vents themselves. Superheated fluids billowing from black smokers emit a faint, radiant glow, a thermal radiation from waters exceeding 350 degrees Celsius. To a human eye, this light would be imperceptible, but the shrimp's dorsal organ is sensitive enough to detect it. This biological innovation allows the shrimp to navigate the razor's edge of its habitat, steering towards the life-giving chemical soup while avoiding the lethal, superheated outflow just a few centimeters away. This navigation is writ large in the swarming behavior that defines the species. Around the mineral spires of black smokers, Rimicaris exoculata aggregates in densities that challenge comprehension. Tens of thousands of individuals packed into a single square meter. The shrimp form a living, pulsating carpet that undulates with the rhythm of the currents, a dense, coordinated mass that moves as one. Each individual is locked in a delicate dance, using its photoreceptive organ to maintain perfect position within the thermal and chemical gradient of the vent fluid. Remarkably, the shrimp do not simply feed on the vent's bounty directly. They are meticulous farmers, cultivating lush gardens of symbiotic bacteria on their own bodies. The inner surfaces of their gill chambers, housed within the carapace, are lined with a dense felt of filamentous bacteria. The shrimp tend these crops by continuously passing oxygenated water over their gills, sweeping their specialized mouthparts across the bacterial mats to harvest the microbial growth. It is a self-contained and mobile farm, a symbiotic cycle that sustains the immense swarms. In the absolute darkness of the deep ocean, Rimicaris exoculata has woven together a suite of adaptations that feel almost visionary. A degenerate eye reborn as a thermal light detector, a societal swarm navigating by the invisible glow of hydrothermal radiance, and a body transformed into a mobile garden all converge on a single, elegant solution for survival. The shrimp stands as a quiet testament to the profound creativity of evolution, a creature that has found its way not by the light of the sun, but by the faint whisper of light from the earth itself. The discovery of hydrothermal vents in 1977 along the Galapagos Rift shattered our understanding of where life could thrive. But it was the systematic exploration that followed which revealed the true scale of this hidden realm. By 1984, a team aboard the submersible Alvin located the first vents on the Juan de Fuca Ridge off the coast of the Pacific Northwest. Here, towering mineral spires and dense clusters of tube worms proved that the Galapagos site was not an isolated miracle, but part of a larger phenomenon waiting to be uncovered. Attention soon turned further south along the East Pacific Rise, where spectacular black smokers belched superheated fluid, and then across to the vast, slow-spreading mid-Atlantic ridge. In the mid-1980s, the first Atlantic vents were found at the TAG hydrothermal field, and they presented a completely different biological cast. Gone were the giant tube worms of the Pacific. In their place were immense swarms of blind shrimp, jostling on the chimney walls. Each new ocean basin seemed to host its own distinct evolutionary experiment. The search pressed into the Indian Ocean, a long-standing gap in the global map of vent systems. In 2001, the Kairai vent field was located on the central Indian Ridge, and its fauna confounded expectations. Scientists encountered the scaly-foot gastropod, a snail that builds its shell with iron sulfides, and yet more species of shrimp, mussels, and anemones found nowhere else. The Indian Ocean vents were not simply a mix of Atlantic and Pacific lineages. They were a unique biogeographic province, raising profound questions about how life had arrived there. For decades, the icy southern ocean encircling Antarctica remained a final frontier, its deep-sea ridges hidden beneath some of the roughest seas on Earth. In 2010, a remotely operated vehicle finally descended onto the East Scotia Ridge and revealed a world of ethereal pale octopuses and dense colonies of hairy yeti crabs. These crabs, farming bacteria on their bristly claws, were entirely new to science, and the community structure was unlike anything seen in the Pacific, Atlantic, or Indian Oceans. The discovery completed the circumnavigation of the globe, confirming that vents ring the planet. Meanwhile, the hunt had moved to the far north. In 2008, the Loki's Castle vent field was discovered on the Mons Ridge, deep within the Arctic Circle between Greenland and Norway. Its 5 black smokers rise from an ancient lava flow, and their mineral-rich plumes support a shimmering microbial garden. The fauna included a new species of tube worm and a distinctive amphipod, yet there were also familiar elements hinting at a deep connection between the Arctic and the rest of the Atlantic Ridge system. This patchwork of isolated deep-sea oases presents a stubborn biogeographic puzzle. The species at each vent field are often endemic, yet their larvae must somehow drift across vast stretches of cold, barren seafloor to colonize new sites. The ridges themselves are broken by transform faults, fracture zones, and entire ocean basins, so the journey can take decades. Currents, whale falls, and even sunken wood may act as stepping stones, but the precise pathways remain largely hidden, a gentle reminder that the deep ocean still holds its most intimate secrets close. In the perpetual darkness of the Pacific Ocean, over 2,000 meters beneath the surface, a remotely operated vehicle illuminated a creature that seemed to belong to mythology. The year was 2005, and scientists were exploring hydrothermal vents near Easter Island when they encountered a pale, long-armed crab with a dense covering of filamentous bristles. They named it Kiwa hirsuta, after the Polynesian goddess of shellfish, and its appearance immediately earned it the common name yeti crab. The most striking feature of this blind decapod was the thick mat of setae, or bristles, that cloaked its claws and arms. A debate soon emerged among biologists about the primary function of these silky filaments. Some evidence suggested they served as a garden, cultivating dense colonies of chemosynthetic bacteria that the crab would then comb off and eat. Others proposed that the elaborate setae were primarily sensory organs, allowing the crab to detect subtle chemical gradients or temperature shifts in the turbulent vent water. The discovery of Kiwa hirsuta was not an isolated curiosity. In the years that followed, researchers discovered 3 additional species of yeti crab, each occupying a distinct niche in the deep ocean's chemosynthetic ecosystems. Kiwa puravita was found waving its claws over methane seeps off the coast of Costa Rica, a rhythmic motion thought to nourish its bacterial crop. 2 more species, Kiwa araunii and Kiwa tyleri, were later documented on the remote ridges of the Southern Ocean, expanding the family's known range from the tropics to the polar deep. Life at the Antarctic vents demands extreme resilience, and Kiwa tyleri has evolved a strategy of astonishing proximity to its energy source. Here, on the sheer rock faces of the East Scotia Ridge, these crabs aggregate in densities reaching 700 individuals per square meter. They crowd so tightly around the scalding fluid that their carapaces often touch, a desperate mosaic of life clinging to the narrow boundary between freezing abyssal water and superheated vent effluent. With the discovery of these 4 species, taxonomists established an entirely new family of crustaceans, the Kiwidae. This family is remarkable not only for its recent recognition, but also for its exclusive association with the chemosynthetic habitats of deep sea vents and cold seeps. Every member of the Kiwidae known to science has been plucked from these island-like oases of chemical energy, a lineage written into existence by the planet's inner heat. The Yeti crab remains a vivid emblem of the deep ocean's hidden biodiversity, its story still unfolding with each expedition. Its hairy arms, whether they are tuned for farming or for feeling, represent a sensory and nutritional adaptation that we are only beginning to understand. In the silent, crushing depths, the crabs continue their slow dance, their pale bodies illuminated only by the faint glow of their own volcanic world. In December 2000, the research vessel Atlantis was mapping a submerged mountain far from the mid-Atlantic ridge axis when its cameras came upon a ghostly forest of pale spires. The expedition had not been hunting for hydrothermal vents at all, yet the submersible's lights revealed an entirely new class of deep-sea structure. Serendipity had just unveiled the Lost City hydrothermal field, a discovery that would quietly rewrite our understanding of how life and water can interact in the dark. The most magnificent of its monuments is a towering chimney named Poseidon. Rising 18 stories above the seafloor, it is a creamy monolith of calcium carbonate, not the dark sulfide of typical black smokers. Its flanks are sculpted into fluted columns and delicate ledges, as if a sunken cathedral had been lifting itself stone by stone from the abyss for over 30,000 years. The fluids that seep from these chimneys are barely warm by hydrothermal standards, flowing between 40 and 90 degrees Celsius. They are also profoundly alkaline, reaching a pH of 11, which is closer to household ammonia than to the acid brew of a black smoker. Instead of a torrent of scalding metal-rich water, the Lost City exhales a gentle, hydrogen-rich breath that feels almost hospitable. This gentle chemistry is powered by a process called serpentinization. Seawater percolates deep into the mantle rock, where it reacts with the mineral olivine, transforming it into serpentinite. The reaction generates heat, releases hydrogen and methane, and charges the fluid with the alkalinity that eventually rises through the white chimneys. No volcanic magma is needed to drive this engine, only the restless meeting of rock and ocean. Life here has built a kingdom not on sulfide, but on methane and hydrogen. The towering spires are draped in thick gelatinous biofilms dominated by archaea, organisms from a branch of life rarely seen in such abundance. These microbial communities are entirely different from the tubeworm and mussel colonies that crowd black smokers, and many of the microbes thriving here have never been found anywhere else on Earth. To many researchers, the Lost City feels more like a window into life's beginnings than the superheated acidic vents discovered decades earlier. The warm, alkaline, hydrogen-saturated conditions of its chimneys resemble the kind of chemical gradients that might have nurtured the first self-replicating molecules on a young planet. In this quiet field of white towers, the deep ocean may be offering a glimpse of the cradle from which all life emerged. In the spring of 1991, a volcanic eruption along the East Pacific Rise at 9 degrees north fundamentally reset a patch of the deep ocean floor. A sheet of black lava, over a kilometer wide, flowed across the seabed and buried vibrant hydrothermal vent communities under a glassy, sterile crust. The towering tube worms, swarming crustaceans, and waving anemones that had thrived in the chemical-rich waters were extinguished in an instant, leaving behind a landscape that looked more like a cooled planet than a living ecosystem. When scientists returned to the site in the submersible Alvin just 1 year later, they expected to find a barren, lifeless seafloor. Instead, their lights revealed a world already stirring with the first faint pulse of recolonization. Thin wisps of hydrothermal fluid were already seeping through cracks in the new lava, and on the fresh rock surface, delicate white microbial mats spread like a tentative veil. The sheer speed of life's return so soon after absolute destruction was the first surprise. The very first pioneers were organisms adapted to exploit the empty canvas. Microscopic bacteria formed thick, filamentous mats, metabolizing the hydrogen sulfide in the vent fluids and creating a primary food source. Alongside them drifted in the dandelion siphonophores, small gelatinous animals that look like airborne seeds caught in the current, their tentacles extending to graze on the microbial bloom. These species, transient and opportunistic, were the architects of the initial recovery, preparing the ground for a more complex community. Within 2 years, the white microbial landscape gave way to thickets of the tubeworm Tevnia jericonana, a fast-growing pioneer species that can tolerate the still-shifting chemical conditions. Their slender, pearly tubes soon covered the lava in dense clusters, providing a three-dimensional habitat where other animals could shelter. The Tevnia stage acted as a crucial intermediate chapter, stabilizing the flow of nutrients and creating microhabitats that paved the way for a more iconic resident. By 1995, the giant tube worm Riftia pachiptila had begun to overtake the Tevnia, and within a few short years the site was once again dominated by the familiar red-plumed forests. Crabs, limpets, and swarms of vent shrimp reappeared, weaving a food web as complex as the one that had been buried. A full succession from bare rock to a mature climax community had unfolded in less than a decade, a cycle that would have taken centuries in a terrestrial forest. This rapid rebirth revealed a profound truth about the deep ocean. The species that inhabit these volatile, island-like vents are not fragile prisoners of their habitat, but masters of dispersal and resilience. Their larvae drift unseen across abyssal plains, ready to colonize any new eruption that briefly lights up the darkness. The destructive power of the volcanoes, rather than being an ending, emerged as a necessary engine of renewal, resetting the biological clock and ensuring the continuity of life in one of Earth's most extreme environments. Deep on the mid-Atlantic ridge, where the seafloor splits apart and molten rock rises close to the surface, great fields of hydrothermal vents roar into the abyss. Here, in total darkness and crushing pressure, lives a quiet giant, a mussel known as Bathymodiolus azoricus. It clings to the basalt chimneys and lava flows, bathed in shimmering mineral-rich fluids that would be toxic to most life. Yet this humble bivalve has found a way not merely to survive, but to flourish in one of the most extreme environments on Earth. The secret to its success lies hidden within its gills. Each individual hosts a remarkable dual colony of symbiotic bacteria, two distinct types living side by side inside specialized cells. One group oxidizes hydrogen sulfide, the other consumes methane. Both convert these volcanic chemicals into simple sugars and other nutrients that feed the mussel directly. The animal simply absorbs the gases from the vent fluid through its gill tissues, providing its internal partners with everything they need to power a self-contained food factory. This dual symbiosis grants Bathymodiolus azoricus a rare flexibility. At a black smoker, where fluids are rich in sulfur compounds, the mussel can rely more heavily on its sulfide-oxidizing bacteria. Yet the same species also thrives at cold seeps, where methane bubbles up from decaying organic matter far beneath the seabed. There, the methane-oxidizing symbionts take over the bulk of the workload. This ability to switch metabolic strategies means the mussel can colonize a far wider range of deep-sea oases than any vent creature dependent on a single energy source. Where these mussels settle, they do not arrive alone. Over time, layer upon layer of individuals fuse their tough byssus threads to the rock, building dense three-dimensional beds that can stretch for many meters. These living platforms transform the bare volcanic landscape into a structured habitat. The rough shells and the sheltered spaces between them offer refuge to a host of smaller organisms, from tiny limpets and scale worms to juvenile crabs and shrimps, effectively creating an island of life in the deep desert. The pace of life here is slow and steady. Unlike coastal mussels that mature in a season and rarely live beyond a decade, Bathymodiolus azoricus grows to a remarkable size, its shell often reaching 15 centimeters in length. More astonishing still is its longevity. Scientists have discovered individuals that are well over a century old, their slow, persistent growth recorded in the layers of their shells. They are patient architects, building their communities across generations that span human lifetimes. In the unending night of the deep ocean, these mussel beds become a foundation for an entire ecosystem. They do not simply endure the hostile chemistry of the vents, they transform it into a generous source of sustenance. Through their quiet, filter-free symbiosis, Bathymodiolus mussels reveal how life can rewrite the rules of existence, forging abundance from the raw minerals of the planet itself. In the deep ocean, hydrothermal vents spew a cocktail of chemicals that would be lethal to most forms of life, chief among them hydrogen sulfide. This compound disrupts the very machinery of respiration, binding to the final enzyme in the electron transport chain and suffocating cells from the inside. Yet around these chimneys, life has not simply endured. It has erupted into a dense and improbable ecosystem. The animals here have found a way to neutralize the poison that rains down upon them. The key to their survival lies in an arsenal of specialized blood proteins that intercept and detoxify hydrogen sulfide before it can reach vulnerable mitochondria. Many vent animals, from mussels to clams, possess sulfide-binding proteins that sequester the toxin and transport it safely through the body. These molecules act as a chemical buffer, maintaining a constant, low concentration of free sulfide in the tissues, turning a deadly compound into a usable fuel for their symbiotic partners. The giant tube worm, Riftia pachyptyla, has refined this strategy to a biochemical marvel. Its blood contains an enormous extracellular hemoglobin molecule that solves a seemingly impossible problem, carrying both oxygen and sulfide simultaneously without the two reacting. The hemoglobin binds oxygen at a set of hemisites, while sulfide attaches to completely different, specially positioned zinc atoms. This dual transport system delivers the two essential ingredients to symbiotic bacteria living deep inside the worm's body, where they are combined to power a living factory in the complete absence of sunlight. Life on the chimney wall is a constant negotiation with temperature. The shimmering fluid erupting from the vent can exceed 350 degrees Celsius, and a direct hit would cook any animal in a fraction of a second. Vent organisms avoid this scalding core by clinging to the precise boundary where superheated water mixes with the near-freezing deep sea. They read the turbulence and the thermal gradients, colonizing the substrate just centimeters from the plume, a world defined by a sharp and invisible frontier. Different species exhibit remarkable and distinct thermal tolerances. The Pompeii worm, Alvanella pompeiana, endures one of the most extreme lifestyles, living with its head in water of a moderate 20 degrees Celsius, while its tail end, nestled inside a papery tube, can experience brief spikes up to 80 degrees. Bythigrade crabs patrol the warm rocks, but painstakingly avoid the fluid's lethal core, while swarms of shrimp, Rimicaris exoculata, jostle for position in the turbulent eddies of the mixing zone. Their bodies adapted to a narrow thermal window that is no more than a few degrees wide. This habitable band, a thin ribbon of just a few centimeters where temperature and chemistry align, is one of the most fiercely contested pieces of real estate on the planet. Space here is the ultimate currency, and the fallout from each new chimney collapse sparks a slow-motion war for dominance. Different species overgrow, nudge, and dislodge their neighbors, jostling for the perfect spot, close enough to the vent's chemical riches to thrive, but far enough to avoid being boiled alive. In the absolute darkness of the deep ocean floor, new hydrothermal vents are born in silence, and the first life to arrive is invisible. Microbial mats, delicate films of bacteria and archaea, drift in the current and settle on the fresh basalt. They begin to weave a living blanket over the scalding rock, tapping into the chemical riches pouring from the vent fluids. These pioneers are the true ecosystem engineers, transforming a sterile mineral landscape into a habitat capable of supporting a cascade of other organisms. The diversity hidden within these mats is staggering. Genetic surveys of hydrothermal vent communities have revealed a multitude of archaea and bacteria, many of which belong to phyla entirely unknown to science before the exploration of the deep sea. These are not just new species, but entire new branches on the tree of life, lineages that diverged billions of years ago and have remained hidden in the planet's thermal engine rooms. Their existence rewrites the catalog of life's forms and biochemical capabilities. Among the most remarkable adaptations is life at temperatures that would instantly sterilize the surface world. Thermophilic microbes thrive at 80 degrees Celsius, while their hyperthermophilic relatives push the boundary even further, growing comfortably between 100 and 121 degrees Celsius. In these scalding waters, where the pressure prevents boiling, proteins and membranes have evolved extraordinary stability. The enzymes they possess, heat-stable and resilient, have become invaluable tools for biotechnology and offer a glimpse into the chemistry of life at its absolute limits. One hyperthermophile has come to symbolize this resilience. Pyrolobus fumari, an archaeon first isolated from a black smoker wall, holds its place in the record books by growing at temperatures up to 113 degrees Celsius. It is a creature of the extreme, its cellular machinery perfectly tuned to the superheated fluids of the vent. Remarkably, it can even survive being heated to 121 degrees Celsius for extended periods, a temperature used in autoclaves to kill all other known life. Pyrolobus fumari does not merely endure the vent, it requires it, ceasing to grow when the temperature drops below a still blistering 90 degrees. The study of vent microbes has fundamentally altered our understanding of the tree of life. Before the discovery of these ecosystems, the living world was neatly divided into plants, animals, fungi, and bacteria. The isolation of extremophilic archaea from vents forced a complete revision, revealing that archaea are a distinct domain of life, as different from bacteria as bacteria are from us. The deep branches of the tree are now populated by vent organisms, including the Asgard archaea, a group that holds clues to the very origin of complex cells. The vent is no longer just a biological curiosity, it is a window into the deepest chapters of life's history on Earth. As ecosystem engineers, these microbes do more than simply colonize. They precipitate minerals, stabilize chimney structures, and fix carbon dioxide into organic matter, forming the base of a food web that sustains giant tube worms, clams, and blind shrimp. They are the hidden architects of a world unlit by the sun, a world driven entirely by geochemical energy. In the quiet, crushing darkness, these invisible communities continue to shape the planet just as they have done for over 4 billion years. In the cold darkness of the deep ocean, where sunlight has never reached, hydrothermal vents tear open the seafloor and release superheated water rich in dissolved minerals. Each new vent field discovered has forced scientists to redraw the known boundaries of the biosphere, expanding it downward into the crust and outward into environments once considered sterile. What began as a curiosity about a few hot springs has become a systematic revision of where life can exist. Before vent research, biologists believed that life required a narrow range of temperatures, perhaps up to 80 degrees Celsius for the hardiest microbes. Vent organisms obliterated that assumption. The Archeon Methanopyrus candleri was observed growing at a 122 degrees Celsius under high pressure, setting a biological record that has not been broken. This single species pushed the thermal ceiling of life by more than 40 degrees beyond what was previously thought possible. Pressure tolerance was redefined in the same way. Vent communities thrive at pressures exceeding 400 atmospheres, depths that would crush conventional laboratory equipment. The enzymes of these organisms, known as piezophiles, function optimally only under such crushing force, revealing that pressure is not a barrier but a requirement for many forms of life. The deep biosphere, once considered empty, now appears to be the largest microbial habitat on Earth. Chemical tolerance underwent an even more dramatic redefinition. Vent fluids are laden with hydrogen sulfide, heavy metals, and acidity that would kill most surface organisms. Yet the extremophiles that colonize these chimneys not only survive but depend on these toxins for energy, using chemosynthesis instead of photosynthesis. They have turned the most hostile chemical environments into thriving ecosystems, proving that life can be fueled by the planet's own internal chemistry. The philosophical shift has been profound. Life was long understood as a surface phenomenon, dependent on sunlight and a thin veneer of temperate conditions. Vent research has transformed that view into something far larger, life as a planetary phenomenon, capable of occupying the deep crust, the ocean floor, and even the subsurface of other worlds. The discovery of extremophiles in vents suggests that if life exists elsewhere in the solar system, it may well be found in similar dark, high-pressure, chemically rich environments. Each new vent brings a new organism, a new tolerance, a new limit pushed further outward. The biosphere is not a thin shell around the planet, but a complex three-dimensional volume that extends miles into the rock and sediment. Vent research has taught us that the definition of habitable is not fixed by Earth's surface conditions, but by the versatility of life itself, a lesson that reshapes how we search for life beyond our own world. In the cold, dark waters 2.5 kilometers below the surface, the submersible Alvin offered a rare window into the deep ocean. Among its pilots was Cindy Lee Van Dover, a biologist who became the only person to both command the vessel and conduct scientific research on hydrothermal vents from inside its titanium sphere. Her work transformed our understanding of how life thrives in extreme environments, from the bizarre tube worms of the East Pacific Rise to the chemical basis of entire ecosystems independent of sunlight. The story of vent discovery itself began with a trail of clues across the seafloor. In 1976, Robert Ballard and his team deployed the Angus camera sled, a deep-towed platform that captured thousands of photographs. Among the images of bare rock and sediment, one frame revealed a startling cluster of clamshells, hinting at a hidden biological oasis. That photograph set the stage for the historic 1977 dive on the Galapagos Rift, where scientists first laid eyes on hydrothermal vents. Alongside the submersible explorations, a different kind of survey was unfolding from the surface. Kathleen Crane, a marine geophysicist, developed methods to detect hydrothermal plumes using temperature and chemical sensors towed behind ships. Her work mapped the invisible signatures of vent fields across vast stretches of ocean, revealing that these chimneys were not isolated wonders, but part of a global network of seafloor spreading and fluid circulation. Decades later, a new kind of vent field emerged from the depths of the Atlantic. Deborah Kelly, a professor at the University of Washington, led the team that discovered the Lost City hydrothermal field in 2000. Unlike the black smokers of the Pacific, Lost City is built by a different chemistry, one driven by the reaction of seawater with mantle rocks, producing towering white carbonate spires and a unique ecosystem that may resemble the earliest life on Earth. The discoveries of these scientists and their colleagues turned the study of hydrothermal vents into one of the most active and internationally collaborative fields in ocean science. Research vessels from the United States, Japan, France, Germany, and many other nations now share data, submersibles, and expertise. The vents themselves, straddling international waters, have become a natural laboratory for understanding the boundaries of life, the origins of ocean chemistry, and even the potential for life on other worlds. From the first glimpse of clamshells in a photograph, to the mapping of invisible plumes and the exploration of alien landscapes like Lost City, the career of each scientist built upon the work of others. The deep sea, once considered a barren desert, has revealed itself as a dynamic and interconnected realm. And the questions raised by these discoveries continue to drive expeditions, inviting new generations of researchers to probe the vents and the secrets they hold. The vents continue regardless. The black plumes rise from the seafloor with the same force they have had for billions of years, indifferent to the submersibles that occasionally descend to watch them. The tube worms extend their red plumes into the current. The shrimp cluster near the heat. The bacteria fix their molecules of sulfur and water into the slow, patient architecture of life. What was found on February 17, 1977, was not just a new ecosystem. It was proof that life does not need permission from the sun. It finds its heat where it can, and it holds on. Let that knowledge settle into the dark with you now. The deep is patient. So is the night.